Hi everyone, welcome to this HBI 101 series talk, Ion Channels 101. It is my pleasure to introduce Professor Bruce Bean, who's a professor of neurobiology that many of you know here in the neurobiology department. He grew up in Schenectady, New York, did his undergraduate work here at Harvard College, then served as a lab tech in cardiology here at Boston Children's Hospital. He did his PhD in biophysics at the University of Rochester, where he worked with Peter Schrager on the pharmacology of sodium channels in the giant axons of crayfish. Then he went to Yale to do a postdoc in cardiac electrophysiology with Richard Chen, and has served as a faculty member at the University of Iowa and the Wilhelm Institute OHSU, before coming to Harvard. His current research focuses on the diversity of different ion channels in mammalian neurons of different types. And he is interested in the therapeutic applications of this for many different diseases, including pain and epilepsy. And a little housekeeping note, if you have questions in Zoom, of course, you can type them into the chat at any time. And if you are signed up for the lunch, please come to the front at the end. And if you are on the waiting list, you're feel free to also come up to see if we have spots. So thank you so much, Bruce.
Thanks everybody for coming. So this is designed as a discussion, and so please interrupt anytime. You know, I brought some pictures and I'll go through and show some pictures, but really I was thinking the most interesting thing would be to just have a discussion of the general topic. So interrupt as we go along. And what I'm going to do is just try to give a sort of broad introduction to ion channels. I'm going to focus on their role in electrical signaling in neurons, but you know, glad to talk about other aspects of ion channels too. I'll touch on a few other aspects. So the essence of the way neurons work is through electrochemistry. And, you know, I've always been struck by, as we all have, how beautiful neurons are, you know, the just unbelievable structure of them. And for people who went to Carl Svoboda's talk on Monday, you know, with these new techniques, one of the most amazing things is being able to follow single axons, you know, throughout the brain. And if you saw his talk, it was phenomenal. I mean, these single neurons had axons that branched all over the brain, whereas mostly with, you know, Ramon y Cajal's original pictures and most of them since then, it's kind of hard to get the axon, you know, the full 3D of it. But the way the neurons work is by generating electrical signals, basically integrating electricity in their dendritic tree, converting that to patterns of action potentials in the cell body, and then that pattern of action potentials goes down the axon. Synapses onto other neurons and cites or inhibits them. So what I want to talk about is the basics of the electrical excitability of neurons. And, you know, that's really, I'd say most easily illustrated in a single picture by showing the threshold for an action potential recorded intracellular. So I'll talk a little bit about extracellular, intracellular recording. But here we're recording the voltage inside a cell. This is with a patch pipette that's making contact with the inside of the cell, recording the voltage there. And what neurons do that's so special is that here, if you inject short bursts of current into the cell membrane, this is a whole cell of a cell body, the membrane depolarizes in response to the current injection in a passive way for small current injections. But then you hit this magic voltage, the voltage threshold, and then just with an extra 0.5 millivolt of depolarization, then the cell does this, fires an action potential. It makes this positive feedback, sudden change in voltage. And if you look at an axon, and these recording from the squid giant axon, where these experiments were first done with intracellular recordings, then you see that these action potentials propagate along the axon and then carry the electrical signal along the axon down to the and so, largely what I'm going to talk about is the way that we as a field understood the machinery behind the action potential in terms of ion channels, and then I'll discuss some other aspects of ion channels also. Okay, so I'm going to go kind of quickly through this, the early part, but I love this, so it's really worth reading if you haven't. The discovery, so electricity was kind of understood, by the Greeks. You could rub amber and see these electrostatic effects. And people got fascinated by this in the mid-1700s. And this always amazed me. Science went really quickly here for about 30 or 40 years because of people like Benjamin Franklin. Did experiments, wrote letters to each other, published, and it very quickly, from like the 1740s to the late 1780s, there was a huge amount of progress made in understanding electricity. And so they would, people devised these machines where you could charge up with static electricity huge amounts, and then you could go, shock each other. And this became, very popular in sort of the French aristocracy, where people would do these experiments as sort of, after dinner entertainment. And the kind of unscientific understanding of it was greatly advanced by the invention of the Leyden jar, which was a way of storing the electric charge. And so you could charge up these Leyden jars. And what a Leyden jar is conducting material surrounded by an insulator with another conductor on the outside. And so what you can do is, using a metal conductor, you can generate static electricity, build it up, and build up a huge charge across the insulator here. So one of Benjamin Franklin's friends in England sent him a Leyden jar, and he became obsessed by it and started doing experiments for like immediately he wrote this letter saying, ever since I got the Leyden jar, I can't stop doing experiments with it. And so one of the things he did was to, and so they, with this, people started to think about electricity in a scientific way. And one of the great things before I get to Benjamin Franklin is one of the great experiments was done by this Abbe Nollet, who decided to see how fast this electricity would conduct. And so because he was abbot of this monastery, he could tell everybody what to do. So he lined up 200 monks, and I just did the calculation this morning. It could not have been a mile long. It doesn't work. You would need 880 monks to do a mile. So it's a little bit of an exaggeration. And then they held on to. They knew that electricity was transmitted through iron wires, and so they hooked up this, and then he took the two people on the end of it, hooked them up to a Leyden jar, and so everybody jumped at the same time, and so concluded that this, whatever this was, it moved incredibly quickly. So oops, I missed. Oh goodness, let's see. I guess I missed. Well, what Franklin did was to discover that he could take apart a Leyden jar. He discovered that he could charge up the material here and then take the whole thing apart. And as long as the charged part didn't touch anything, he could put it back together again. And he was the person who really worked out he invented plus and minus for electric charge, had this theory that there were these two polarities and that a current would flow whenever there was a separation of these charges. And then it was Galvani, not much later, really only 10 years or so later, who got the idea that electricity was somehow involved in animals, and found that if he touched iron pieces of iron to frog legs, that they would sometimes twitch. And so then he got obsessed by doing these experiments. He had a laboratory in his house. He worked very closely with his wife, and he would do things like, so Benjamin Franklin had realized, as we all know from the story of his kite, that he hypothesized that lightning was the same basic phenomenon as the charge in a Leyden jar, and that you could make sparks from both ways. So one of the things Galvani did was to hook up an antenna so that he could get charge from the atmosphere, and then show that if he connected it with a wire to a frog leg, he could make it twitch. And so then he also would connect nerves, especially cut nerves, through an iron wire to the frog leg, and then make the frog leg twitch. So he formulated this idea that nerves and muscle worked by electricity. And then there followed a really interesting long dispute between Galvani and Volta, who was a little bit younger, who started by repeating Galvani's phenomenology, but didn't believe in the interpretation. And so, of course, Volta was the person who discovered that you could make powerful batteries, so you didn't have to just charge up static electricity to get a source of electricity, but you could have dissimilar metals with salt solution and make large voltages. And so he used this to do experiments. And they had this long-running dispute where they were both right and both wrong, where Galvani had concluded that nerve and muscle, carry electricity, that they use electricity in the nerves to cause a muscle contraction. Volta did not believe this. He thought that in all Galvani's experiments, all he was doing was basically making a battery of some kind with the iron that he was using, and that was making the muscle twitch, but not because the muscle itself involved electricity. And so he was actually right about that. And on the other hand, Galvani was obviously completely correct that electricity was used by nerve and muscle in a native way, but he thought this was completely different than the electricity of lightning. And it's, by the way, Galvani's experiments that led to Mary Shelley's story of Frankenstein, because Galvani would go around and give these demonstrations and talk about how living beings used electricity to be alive. And then, she extended that with the idea that you could revive a corpse using electricity. Okay, so now let's get into the science here. So, all right, so the essence of the way neurons work, and actually all biological cells, is that they're basically like little Leyden jars, because they are conductors made by salt water, salty solutions inside and outside, separated by an insulator, which is the biological membrane. And so you can charge up the voltage inside the cell across the membrane. And the reason that happens is that the membrane, and this was understood pretty early in thinking about nerves and other kinds of cells, that there was a voltage inside the cell. This was kind of hypothesized. It was hard to measure directly. Couldn't at this point, do a direct measurement of the voltage inside the cell. This was inferred from various other experiments, and people, then realized that the potassium inside cells was high compared with extracellular, and the sodium was low. And then realized that you could explain, you could produce a voltage inside a cell if there was selective permeability to potassium in the membrane. Because then what happens is potassium tends to go down its concentration gradient if the membrane is selectively permeable to potassium. And it will stop when the voltage inside becomes negative enough to retard its exit. So that's how you generate a resting potential in a cell. And most cells have a resting potential. Most cells have a resting potential inside, not only neurons and muscle, but most other cells too. Typically about -60 to -90. And it's because at rest, their membrane is selectively permeable to potassium. And just a brief digression. So how do you generate this gradient of potassium and sodium? That's done using energy. That's done using pumps that actively extrude sodium and carry potassium into the cell. So the main pump is the well-known sodium potassium ATPase pump that pumps sodium out, potassium in, and uses energy from ATP to do it. So people for a long time did recordings of action potentials, realizing they propagated along nerves. And basically, they would do recordings from the outside of a nerve and see that there was an electrical signal that propagated. And you can measure the propagation velocity pretty well. And in the early 1900s, people invented, using vacuum tubes, better ways of amplifying voltage signals. And so you could then boost the voltage signals and look at the extracellular voltages with much higher resolution than had been possible before. The oscilloscope was invented, so you could then, amplify the extracellular voltage, display it on an oscilloscope. And so there were a whole bunch of labs. Gasser and Erlanger were two of the main people who worked on this. They would take sciatic nerves, for example, from a rat or a frog, and do an extracellular recording. You could measure conduction velocity by boosting up the electrical, the extracellular signal. But nobody knew what really was happening with the voltage inside this cell. And the key to that was then being able to record the voltage inside a cell directly. And that was first done by Hodgkin and Huxley in 1939, where one of their colleagues, J.Z. Young at Cambridge, who was interested in the nervous systems of many kinds of animals, discovered that the squid has an unusually large axon with an unusually large diameter. And the reason the squid has that is that the conduction velocity of an unmyelinated nerve is faster the larger the nerve is. And so if you need a nerve to conduct really quickly, it should be very big. And the squid has evolved this giant axon, which mediates an escape reflex, where anything it immediately jets away in response to any disturbance. And so J.Z. Young had told them about this unusually giant axon with the idea that maybe that would be useful for studying electrophysiology. And so what they did was to dissect out the axon, hang it so it was straight, and then put a fine pipette down the axis of the axon, far enough away from where the axon was cut, so that it was no longer, the voltage was no longer affected by the cut part. And when they did that, as they expected, they saw that the inside of the cell was negative. And then if they put an electric shock on part of the axon, did the recording, the surprise was that the voltage actually went positive. Most people at the time thought that what would happen is that when the action potential was generated, that the permeability to potassium would dissipate and the thing would become sort of generally permeable to everything and the voltage would go to zero. Instead, they found that it actually went above zero. And like with a lot of things, there were all sorts of controversy about this. A lot of people didn't believe it. They thought it was an artifact of the, of the electrodes that they used to do the recording and so forth. But it was true. And so then, there was a little gap during World War II where they were working on radar and couldn't do experiments anymore. But then when they came back and started doing more experiments on the squid axon after the war, they did an amazing series of experiments, which are encapsulated in the famous Hodgkin-Huxley papers of 1951, and very well worth reading to this day. There's a fantastic book that came out about a year ago by Indira Raman and David Furster, which is an annotated version of the Hodgkin-Huxley papers, where they sort of go through the history of what they were doing and sort of unpack each experiment in terms of what the logic was, what they learned from it. And those papers culminate in what I think is really the first paper of computational biology, where Hodgkin and Huxley made equations based on the way the ionic conductances in the membrane worked according to their experiments, and then could reconstruct exactly the way the action potential, the shape and the properties and the conduction velocity of the action. It's just a phenomenal series of papers, very well worth reading. And so what they discovered is that the way the action potential works is that at rest, there's selective permeability to potassium from some sort of resting potassium channels in the membrane. And that the action potential is generated by a huge increase in the sodium conductance. An enormous increase that happens very quickly, hundreds of microseconds, and is transient. So then, as they called it, inactivates after a millisecond or so. So the action potential is generated extremely quickly because of a huge sodium permeability. When the membrane becomes permeable to sodium ions, sodium is high outside, low inside. Sodium comes into the cell, and those sodium ions cause a depolarization. And they cause a depolarization until the inside of the cell becomes so positive that sodium no longer comes in. So that's why the voltage goes so positive during the. And they found that there was also another kind of potassium conductance in the cell that was activated later, that was activated by depolarization, and that helped in the depolarization of the action potential. And that potassium conductance produces what they recorded as an after hyperpolarization, where the membrane potential transiently becomes more negative after the action potential, because the potassium conductance goes up even higher than it was in control before the action potential. So they understood this. They were not thinking about ion channels. They were not thinking about pores in the membrane. They were thinking about that these voltage-dependent conductances. So they didn't talk about channels. They talked about these as conductances. And they were very sort of careful in their discussion not to say much about it. And apparently they were thinking of carriers in the membrane. They were thinking of that these probably worked as something where sodium would bind to something and then be transported through the membrane in a complex with it. And really the next series of experiment, the next sort of idea that these conductances involved in the action potential were from pores in the membrane, were experiments done by Clay Armstrong and Bernal Hill in the late 1960s and the early 1970s. And so they did, I'll go through very quickly some of their experiments because I just love these. I was lucky enough to be an undergraduate. I was a miserable undergraduate, and miserable in every sense of the word. Miserable student, miserable in my life. But the one good thing was my senior year, my junior year, I was studying philosophy and I hated it. to the seminar in Descartes, and I couldn't believe it. It was like a hot day, and this, graduate student was droning on and on about these Descartes essays that we wrote. And I thought, why am I listening to what somebody, in the 1600s thought about how the mind worked? I mean, certainly we've learned something since then. And so I transferred to biology and started taking some biology classes. And the spring of my senior year, there was a series of guest lectures, 3 guest lectures. Mike Bennett talking about electrical synapses, and Walter Hagen's talking about the ear, and Bertil Hille, who was talking about ion channels. And so he and Clay Armstrong had developed the idea, which many people didn't accept, that these conductances, these voltage-dependent conductances, were from little pores in the membrane, little protein pores. And so I'll show you some of the experiments that led them to hypothesize this. So this is the basic kind of experiment that you do. So this is the kind of experiment Hodgkin and Huxley did, where because these are voltage-dependent events, the key was to control the voltage across this cell. So people actually in the US, Kenneth Cole and Howard Curtis, had worked out a way of doing this, although they never really pursued it, themselves all that effectively. But Hodgkin learned about this and then set up a system to do this. So this is the voltage clamp, where you can clamp the voltage across the membrane anywhere you want. Use a positive feedback circuit to do that. And then you can measure the currents that go through the membrane. And that was the key, because these conductances are voltage. And so by controlling the voltage, you can see exactly what they do. So this is one of the records from Hodgkin and Huxley in the squid axon. What they saw was, at rest, there's basically no conductance. And if you depolarize to a certain point, say here, it's minus 10 millivolts, then you have an inward current followed by an outward current. And they hypothesized, and they did some experiments to convince themselves that this inward current was carried by sodium coming in through the membrane when the membrane was depolarized. The outward current was a slightly delayed conductance that was potassium going out through the membrane, carrying it out. And so when Hille was a graduate student at Rockefeller University, he began doing similar experiments using, in this case, a frog node, another sort of axon that you can voltage clamp in various ways. And he started doing pharmacology. And so Toshio Narahashi had discovered shortly before that the poison in pufferfish was blocking nerve fibers, and that there was this alkaloid, tetrodotoxin, that if you purified it and you put on a nerve fiber, and Toshio and Bertil did this about the same time, you could selectively inhibit the inward current. And so it's as if, leading to the idea that these are really 2 separate things, the flow of the inward sodium current and the flow of the outward potassium, which is unaffected by tetrodotoxin. And then Hille discovered that you could use this simple molecule, tetraethylammonium. So it's just a nitrogen with 4 ethyl groups off it, positively charged ion, a cation. And it happens that if you put it on the outside of a frog node, it inhibits the potassium. And so he began doing experiments with these and sort of thinking about how these were working. And at the same time, Clay Armstrong, who was a medical student who decided he didn't want to do medicine, he wanted to do research, and he was working in Kenneth Cole's lab at NIH, and began doing experiments on squid giant axons, also kind of using pharmacology in a very clever way. And so Armstrong, actually, by the time he was doing these experiments, he had moved to the University of Rochester and worked with some chemists at Kodak to make a series of derivatives of ammonium ions, and made some that had, so this is like tetraethylammonium, except it's got a 9 carbon chain added to it. And he found that when he made these, they didn't do anything from the outside of the squid axon at all. But if he put them inside the cell, then he would get this time-dependent, as he called it, block of the potassium conductance. And he began to imagine that the way to explain this was in terms of an ion channel. And he did an incredibly clever series of experiments. I mean, also really guided by intuition, and ended up, coming up with this idea for what was happening when he put these inhibitors inside the axon. You can't see anything. I mean, the only thing he's recording are these things on an oscilloscope, and then imagining what must be going on. That's why I just love these experiments. There's nothing. It's all imagination. And so he drew these pictures of what he thought was going on that turned out to be exactly correct, when the structures of these were worked out in 1998 by Rod McKinnon. And so one of the ideas he had was, well, maybe the way the potassium channel works to select potassium over other ions like sodium ions, he formulated the idea that normally potassium is surrounded by waters. It's hydrated because it's positively charged and interacts with the electronegative parts of the water molecule. And that, roughly speaking, it might have like 4 water molecules around when it's hydrated. And so then he formulated the idea that maybe what happened inside the channel to allow the movement of potassium was that there would be a narrow part of the channel where the potassium ion would become dehydrated and the water molecules would essentially be mimicked by something in the channel. And so they start to think about, well, this makes sense if you had a channel to do this, not a carrier. You could really explain this by a channel. And then he had the idea, and the reason he made this molecule was he was thinking, maybe I can make something that looks a little bit like a hydrated potassium ion, a similar size, but a little bigger, so that it won't go through the channel because it can't get dehydrated. And so what he found was, when he put these charged cations, these sort of big charged cations inside, that his hypothesis was that this blocking was because they would go into the channel, but they wouldn't conduct, and they would block the movement of potassium ions. And one of the coolest experiments he did here was he found that if he had one of these inside, the channel opened, the blocker came in, and then he repolarized the membrane where the channel would close again. It didn't close right away. It conducted for a little while, and he concluded that's because there was a gate on the inside of the channel that could not close when this blocker was inside. And so it would have to wait for the blocker to come out before the channel could close, and that's why he saw this inward current that was flowing as the blocker came out and the channel conducted. I mean, it's amazing. It was all imagined, right? But it was correct. And he convinced himself it could only work if it were a channel, and everything made sense. When he started thinking about the way the channels, he was working with Kenneth Cole, who hated the idea of channels. He didn't believe it. He thought, I don't know what he thought. He did not believe in protein channels being the basis of this. So I remember hearing that place where I had sort of a difficult time as a postdoc, doing these experiments, getting excited about it, and supposed mentor, having interested in this. And so Hilla did these experiments also primarily with pharmacology, and came up with his own sort of, imagined view of what a channel might be like. He also, in various kind of similar experiments, this is a sodium channel, came up with the idea that the gate of the channel must be on the inside of the membrane. And there were a variety of reasons for that. Came up with the idea that in that case, if the gate that controls whether the channel is open or not is on the inside, that maybe the selectivity for sodium ions over potassium ions would be on the outside of the membrane. And part of the idea was that the tetrodotoxin, which binds only on the outside of the cell, has a guanidinium. And Hilla thought, well, maybe the guanidinium group looks a little bit like a sodium ion when it first comes into the channel. Okay, so the way the voltage dependence of the channels works, we still don't totally understand. But the basic idea is that there's a voltage sensor inside the channel. There's a charged region of the channel that is positive charges. They're in the membrane. And when there's a depolarization across the membrane, then these positive charges move from a more inward position to a more outward position. And when they do that, they are coupled to these gates that Armstrong and Hilla hypothesized were on the inside of the channel, which turned out to be exactly correct. And so then the outward movement of these sensors, these voltage sensor residues, which are, we now know, lysine and arginine residues, positively charged residues, that these move within the membrane field from an inward position to an outward position. This was all hypothetical at this point, but validated by later structure. That then it causes the gate in the inside of the channel to open. And it's, functionally actually very similar to a transistor. Where a transistor, the way it works is you put a controlling voltage across it, and then that induces a huge conductance change, a huge resistance change across the transistor. And in fact, I like this figure by Fred Sigworth in the early days of looking at how voltage-dependent channels work, of comparing the voltage sensitivity of a typical transistor with a typical ion channel. And you can see just how incredibly sensitive to voltage the gating of the channel is. It's just with a 10 or 20 millivolts is all it takes to go from being almost completely closed, almost completely open. Okay, so the way the action potential works then, sort of the understanding at this point, this was still sort of cartoons of how the imagined channels would work. But it was very clear that very steeply voltage dependent. And so the way the action potential works is if you depolarize just enough to get a few sodium channels, a fraction of the sodium channels open, then what happens? Well, sodium comes in through those channels. That sodium entry depolarizes the membrane. That depolarization of the membrane opens more sodium channels because they're so steeply voltage dependent. And because of the steepness of the voltage dependence and the regenerative nature of this, everything happens in microseconds, tens of microseconds. The sodium comes in, depolarizes, opens more sodium channels. So within tens of microseconds, you can basically open all the sodium channels. And when that happens, the voltage inside the cell goes to near reversal potential for sodium, which is strongly positive. So now let's go to the 1980s. Okay, so everyone was imagining what these channels, might look like, but nobody had a way of really directly visualizing. And two things in the 1980s happened. Again, I feel so lucky to sort of, lived through this, going to research conferences and ion channels, sort of see this all develop, which worked out really well for me because I'm an incredibly slow learner and I need to hear things like 10 times before they sink in. And it was awesome because years and years hear the same thing. It took me forever to sort of learn the amino acid code, and lysines and arginines were positively charged. My lab was here next door to Rob McKinnon's for years while he and Gary Yellen and other people were doing these beautiful mutagenesis experiments. And it took me years to even understand, modifying these amino acids, which were positively charged, which were negatively charged, all this stuff. But anyway, so two things in the 1980s. So one was the invention of the patch clamp by Neuro and Sock. And that transformed everything because up to that point, the main preparations people had to work with to record channels under voltage clamp were the squid giant axon. So you could go to Woods Hole, work on the squid giant axon, or Plymouth in the UK. The frog node that a guy named Frankenheuser worked out a way of voltage clamping that Bertle Hill and other people used to do. But it was really hard to do much of anything with mammalian cells. You could put a sharp microelectrode in them and usually it killed them. And if they lasted, they didn't last very long. You couldn't clamp the voltage in the membrane. You couldn't really see what was going on. But with the voltage clamp technique that Nehr and Sockman developed, you basically take a pipette that's got a tip of maybe 2 or 3 microns. You fire polish it so it's nice and smooth. And what they discovered after a lot of trial and error was you put that onto a cell membrane, you put a little bit of suction on, and it makes an incredibly high resistance seal with a membrane. So high that it actually is almost equivalent to having covalent bonds between the glass and the membrane. Nobody's ever really quite understood, what happens there. Right? You get this giga-ohm seal, this incredibly high resistance seal between the inside of the pipette and the extracellular solution. And one of the most dramatic use of the patch clamp pipette, the patch clamp technique, was to allow recording of single channels. And so you then isolate a very small patch of membrane. And so then you could do an experiment where if you were lucky, you'd get a single channel under the pipette, and you could then see the opening and closing of that single channel, that single protein, in response to changes. And the other thing that it allowed was if you, I didn't make a picture of this, but if you now rupture the membrane under here by putting a strong suction on, then you get electrical continuity between the inside of the pipette and the inside of the cell. And then you can do a recording from the whole cell. And you can do this on small mammalian cells really nicely. And so everybody got excited about this. We all started, getting whatever cells we could get and starting to make recordings. And the other revolution in the 1980s was molecular biology, where people, starting with Shisako Numa, and then Catterall and other people began, Tom Schwartz and Lily Jan began cloning ion channels, reconstituting and expressing the protein, and then being able to study those reconstituted proteins. And so here's, for example, and you could at that time, make guesses from the transmembrane, from the structure of the channel, the amino acid structure. You could make guesses about what pieces were likely to be transmembrane. You could test that by doing things like putting labels on the inside or the outside, and ultimately come up with models for the structure of ion channels. Here's, for example, the voltage-dependent sodium channel. People realized that there was this special transmembrane region. So the voltage-dependent sodium channel is basically 4 pseudo subunits in a single protein. Each pseudo subunit is kind of like the others. And there's this transmembrane region 4 that has lysines and arginines alternating every third residue. And people found that if they mutated these, you would change the voltage dependence of the channel. So concluded correctly that these are the voltage sensors. These are this S4 region moves from an inward position to an outward position when the membrane depolarizes, and that's the gating charge that controls the channel. So with the kind of revolution, both with cloning and of patch clamp, allowing you to record from all kinds of cells that were previously inaccessible, what we discovered was amazing, that there were just a huge number of ion channels in like mammalian neurons. Not only mammalian neurons, but also Drosophila neurons. And this was a shock because everybody had sort of thought, well, squid axon makes an action potential. We understand it. You know, there's two different conductances, two different ion channels, a sodium channel and a potassium channel. That makes electrical signaling. But what people discovered is a really combination of the cloning and then recording from a whole variety of cells is there's a huge variety of voltage dependent ion channels. And I listed here in the neurophysiology class, these are some of the ion channels that were discovered basically during the 1980s from these kinds of recordings from the cloning. And these are ones that are basically present in almost any neuron that you would go into the brain and explore. And it's by no means exhaustive. You know, any particular neuron has lots of other ones too. And I'll just draw attention to the fact that here's only some of the ion channels that are present in the human genome. I meant to look up what the number is. It's very large, hundreds or probably thousands. And I'm not going to talk about it because I'm going to focus on the channels in the membranes of nerves that give electrical signaling. But intracellular organelles also have all kinds of ion channels, very poorly understood. There's only a hardy few people, like Dejan Ren at the University of Pennsylvania. You know, I mean, it's really hard to do because then you have to figure out some way of doing a recording from a piece of membrane from an intracellular organelle. But they have their own sets of ion channels, you know, that regulate movement of ions, and all sorts of things inside. I'm going to focus on that. So one of the kind of consequences also of being able to record from all kinds of neurons was to realize that even though all neurons make action potentials, that actually the shape of the action was a little bit different from neuron to neuron. It's not all just like the squid axon action potential. And even more than that, the pattern of action potentials varies a lot from neuron to neuron. This was largely the work of people like Dr. Yinas and David McCormick and others who went sort of systematically and started to investigate the way individual kinds of neurons work. Here's one of my favorite recordings, kind of illustrating the complexity of the electrical activity of a neuron. This is a recording from a brain slice from a thalamic relay neuron. Yeah, when he was at Yale. He now teaches happiness studies at the University of Oregon. These days, he put microelectrons into neurons. And so this is what one of your thalamic relay neurons does. Basically at night. This is what our thalamic relay neurons do at night. The reason I say at night is that these are very strongly regulated by modulatory transmitters, like serotonin, norepinephrine, acetylcholine. They're all higher during the day and lower at night. And at night, when these modulatory transmitters are sort of decreased, here's what our neurons do. All right, I'm not going to play it, but what they do is they fire spontaneously. They fire these action potentials in a completely spontaneous way. They do it so rhythmically and so powerfully that synaptic input coming into the cell doesn't do anything. It's locked out. And that's why you have these big EEG waves when you're sleeping, and that you're not sensitive to most sensory input because the thalamic relay neurons are just doing their own thing. It's very robust pacemaking activity. When they're exposed to things like norepinephrine, acetylcholine, serotonin, dopamine during the daytime, the membrane depolarizes. And when it depolarizes, it stops this spontaneous activity. And it now goes into a regime where now it's very sensitive to any input synaptic input, or in this case, just injecting a little bit more current here. And then it fires action potentials in a very graded way, in a way that's very sensitive to anything coming in. So nighttime, it sort of locks out the sensory input by its robust activity. So what are the ion channels that do this? You know, you have to have channels that generate this spontaneous activity, which is actually a lot like the heart. You know, this is very similar to pacemaking in the heart, including actually in the channels that are involved. And then, you know, something happens, which normally would be a modulation by norepinephrine and these other modulators, that puts it then in a completely different state, where now it'll fire short action potentials in a very high frequency way. So, you know, so why do we evolve all of these different ion channels instead of just using two the way that squid axon does? And the answer is that most neurons do much more complicated things than the squid axon. The squid axon does one thing: it propagates an action potential really quickly. And a single action potential in the squid axon will initiate an escape reflex. It just needs to be fast. The pattern doesn't really matter. And so it's actually kind of interesting. Rita Goodman did these experiments that I modeling that I very informative, showing that the squid axon was very poor at encoding information as a pattern of action potentials. And so if you inject current into the squid axon, suddenly you hit threshold. And then the frequency of firing is immediately very high. And it doesn't change much when you change the injection of current. Whereas almost any other kind of neuron you would look at is much better at encoding information as a frequency of firing. And so here's a couple of examples. And that's really the reason that we've evolved so many different kinds of voltage-dependent channels. It allows, by having combinations of channels, a huge variety of firing patterns, and then a huge variety of ways that the electrical. And a lot of that variety comes from variation in potassium channels and voltage-dependent potassium channels. So unlike the squid axon that just has one voltage-dependent potassium channels, mammalian neurons have lots. And potassium channels are made up of tetramers of proteins from the same family, and so they can have this heteromeric structure that has enormous variability. And then there are also accessory proteins that control the gating and the voltage dependence in very complicated ways that we're only still sort of working out. Each one of these families has its aficionados, in labs around the world, doing nothing but sort of understanding exactly how those work. Okay, so I'm going to just kind of very briefly talk a little bit about the structure of channels. So before 1998, this was sort of the picture we had of what a potassium channel was. And then this amazing achievement by Rod McKinnon spent years trying to figure out how to crystallize channels in such a way that he could do X-ray crystallography. Finally succeeded in 1998 and came up with the first actual structure of what a channel looked like. This is a potassium channel from a bacterium. He tried all kinds of different bacteria, all sorts of different things. He spent years, got to isolate it from the membrane, you got to grow up enough to get lots of protein. You have to use detergents in such a way that the channel doesn't get degraded, but you can use crystals of it. Finally succeeded. And it was just so exciting, so beautiful to see this, not least because it's exactly the way Clay Armstrong had imagined it, exactly. Where there was this big vestibule inside that a hydrated potassium ion will sit in. And Armstrong's blockers would go in and fit in the inside. That there was clearly a gate, that the gate was at the intracellular region where Armstrong had assumed it was, and that it's basically the helices coming together as Rod said, in like a teepee in the inside, where you could just imagine it could be either open or closed to control the gating. And there were potassium ions in the crystal in this selectivity filter, where they're coordinated by carbonyl groups that basically mimic the hydration of water, exactly the way that Claire Armstrong hypothesized. And so that I really love this. Armstrong, he was so excited about this, Claire Armstrong. I mean, you know, who wouldn't be? You know, what you imagined in your oscilloscope, and it turns out to be exactly the way things really work. And so here's the way that Armstrong had imagined that the potassium channel would discriminate between the potassium ion and the sodium ion. And his idea was that the potassium ion would just fit into the filter of the channel. Now, sodium ion is smaller than a potassium ion. You know, so how can you make a channel that passes potassium but not sodium? This was Armstrong's idea. And then it turns out that's exactly the way the channel works. That there's this region, this filter region, where the potassium ions are just beautifully coordinated by the carbonyl groups, but the sodium ion is smaller, and so it's not as energetically favorable sitting in there. All right, so I was going to talk about some aspects of channel pharmacology, but I'm going to stop because we're at an hour, and maybe we can just have a discussion of anything else that people are interested in. So thanks for coming, and thanks for being on Zoom. Send your questions in.
I have maybe kind of a basic question, or I don't know how basic it is, but when we talk about like NAV channels being selective for sodium because sodium is smaller, is that with or without the hydration shell?
That's without the hydration shell.
Yeah.
Exactly.
So you still have the effect of like sodium is losing its hydration shell as it goes through.
Yes.
But it is also physically smaller, which is like the main selectivity.
Exactly. For sodium channels, it's a little bit easier because sodium is smaller. And so Hill actually had worked out the selectivity of sodium channels by comparing all cations, and then it worked out the idea that in the sodium channel, it's dehydrated in the selectivity filter, but in that case, it's just by size with the dehydrated ion. That's why the channels are sodium selective. Yeah.
CA1 pyramidal cell or cortical cell right before action potential. There's always small depolarization. What is that?
Oh, yes, that's a really good question. This was what was so much fun about, you know, with being able to record from all these cells, you know, in the 1980s. And so people found, for example, like in the squid axon, you have this big after hyperpolarization. Hodgkin-Huxley explained that's because the K channels are activated and they stay activated. In hippocampal neurons and also in cortical pyramidal neurons, you have the opposite. They don't do that. They have an after depolarization, as you pointed out. That's from, it's actually not totally understood. It's partly from calcium channels in the membrane. So there are voltage dependent calcium channels that get open. They close, some of those close kind of slowly. And so they give you a lingering depolarization. And the other reason for that after depolarization is that the action potential fires in the cell body. It actually fires in the initial segment of the axon first, and then the cell body fires. And then there's a big dendritic tree. And so what happens is that the depolarization of the action potential in the cell body depolarizes the dendritic tree. And that depolarization kind of lingers for a few milliseconds and then sort of comes back and produces that after depolarization. Yeah, I was planning to get into it. I didn't time things very well. I could talk for 3 hours, but in synaptic transmission, the release of transmitter is is produced by entry of calcium into the presynaptic gluton. And that entry of calcium is very sensitive to the exact shape of the action potential presynaptic gluton. And that after depolarization may happen there also and help control the release of transmitter. Yeah.
You did a very nice talk about the history of the ion channel research. So in your point of view, so what are some of the outstanding questions nowadays?
Yeah, that's a good, I was thinking, you know, when I was walking over, I was thinking, that's Farizad asked me to talk about it. I didn't. It's a really good question. I mean, there are so many levels. So for one thing, we don't really understand the details of the voltage dependent gating. And the reason is that, so there's a cryo-EM revolution now, you know, where Rod originally had to use X-ray crystallography to do these things. That was incredibly difficult. Now you can do it with cryo-EM. So there's a huge expansion of the structures we have in ion channels. But the thing is that they are all in the absence of voltage. They're all basically with the channel at 0 millivolts. And at 0 millivolts, the voltage sensors are all out. So we have a lot of structures of channels where the charge movement is out. What's been really hard to do is to get structures where the charge movement is in, as it normally is in the resting cell. So we don't have a really good picture of exactly how the movement of the charged part opens the channel. So that's, you know, if you're into the sort of, you know, real molecular detail, that's one of the big questions. And people are starting to make, you know, progress on that, but it's probably a little bit different in different channels. So it's not that easy. Personally, what I love is ion channel pharmacology, because ion channels are the targets of, turn out to be the targets of a lot of clinically used drugs, all discovered before we knew what their targets were. So anti-epileptic drugs turn out to be sodium channel inhibitors. And, you know, so now that we understand what their targets are, now, for example, there are 9 different gene products for sodium channels in the body. And so now we can start to use, you know, the differences of those sodium channels, for example, to be more rational about designing drugs that will affect electrical activity. It isn't that easy because the circuits in the brain are so unbelievably complicated. And so it's really hard to guess, you know, well, if I modify this potassium channel, this particular sodium channel, what is the effect on the brain going to be? It's really hard to do that rationally. It's still very difficult, and even though we know the targets. And so it turns out that the trial and error way that, for example, you know, epileptic drugs like carbamazepine and phenytoin, you know, they were discovered by trial and error. And turns out that that's a good way to do it still, because even though we know their targets, going from the targets to sort of rational design is not easy, but it can be done. And I think, you know, that to me is, you know, one of the sort of frontiers. And that involves also probably a lot of computational modeling, you know, having a really good computational model for what's going to happen if you change a particular ion channel in a particular neuron in the context of the particular circuit. Let's see, what else? I mean, discovering ion channels that are involved in sensing. You know, there's a lot of very recent work, you know, that's been successful there, like piezo channels involved in hair cell movement, but there's a lot more to be discovered there.
So I can read some of the Zoom questions. Since you mentioned piezo channels, a question just came in about that. Thank you for the wonderful talk. The piezo channels fulfill mechanical transduction. How does the activity of these ion channels modulate the information encoded by the nerves, and how are they different in different nerves that have these channels? Can we find a link between the frequency of firing and the frequencies of mechanical stimulus generated by pressure, stretch, hair cells in nerves with the piezo receptors?
That's a great question. That's different in the case of every cell, I would say. So I think the answer is you have to go in and understand in a particular cell, what does the current look like through the transducer channel? And then what is that going to do in terms of activating sodium channels, in terms of modulation by potassium channels to be converted into a pattern of action potentials? It's all going to depend on the details in every cell. So it's hard to give a general answer to that, but I think it's a really interesting thing to study in all neurons that respond to sensing. Even the way that piezo channels are opened and closed turned out to be kind of hard to understand.
So more Zoom questions. Presumably the function of channels depends on both their spatial distribution and local density within cell membranes. How much is understood about that and about feedback mechanisms by which cells fine-tune their firing properties?
Another fantastic question that is, I would say, cutting edge research. The activity of neurons is plastic. Great example is long-term potentiation at synapses that involves insertion and removal of AMPA receptors, glutamate-gated channels from the membrane. And lots of other kinds of plasticity, including changes in expression of voltage-dependent channels. How that works, what are the feedback mechanisms by which a neuron controls the particular expression of its own ion channels is a huge frontier. We understand very little about it. I mean, all I would say is there's a lot of transcription factors, and there's transcription factors can be incredibly complicated and effective in controlling expression of things. Somehow there's feedback. The idea that Eve Marder has been one of the pioneers in trying to understand this in detail, and the basic idea she has, which I think has a lot of evidence for it, is that in the end it's calcium. It's the pattern of calcium coming in with each action potential that is used as a feedback to then somehow control the expression of channels and then to control the electrical activity. But exactly how that happens is, basically not understood.
In terms of small molecule drugs that bind ion channels, do they typically all do generally the same thing? Do they lock the ion channel in the closed state all the time, or kind of what's the purpose of the drugs?
That's another great question. They can do almost everything you can imagine. So some clinically used drugs, for example, work by locking potassium channels in the open state, which is a really effective way at reducing electrical activity. So there's an anti-epileptic drug called retigabine that acts specifically on Kv7 channels to enhance their opening. A lot of like local anesthetics work by locking the channel in the closed state. Or actually, not in the closed state, but the inactivated state in the case of those. So it really varies a lot. And then there are a lot of toxin molecules. A lot of what we learned originally before cloning even was that there are in venoms of snakes and spiders particularly, there's a huge, and cone snails from South China Sea, there's a huge variety of peptides that are incredibly selective for binding in the outside of particular channels and blocking. And so people, you sort of use that. Well, maybe you can make a small molecule that mimics these, and people are really hard on that.
So I think we have to end, but we do have some more questions in the chat. So if you still have your question and you email it to me, I can forward it. So thank you very much for attending, and thank you so much, Bruce, for the amazing talk.